Okay, let's talk about a prototypical human cell, or a cell. Now there's animal cells and there's plant cells, of course. This is not biology, so we won't study plant cells, but the main difference between a eukaryotic and a prokaryotic, or a plant cell and an animal cell, is that plant cells usually have a cell wall and chloroplast, so plant cells can usually make their own food, whereas we, animal cells, cannot make our own food, and we don't have a cell wall. We do have a plasma membrane, and then we have all these little organelles that we'll talk about in just a little bit here. Alright, so what are these little organelles? Well, organelles really means little organs. They're metabolically active. They compartmentalize contents of the cell. Right, so put them in little compartments. Well, what are the organelles? You've got the nucleus, the endoplasmic reticulum, the ribosomes, Golgi complex, proteasomes, lysosomes, peroxisomes, mitochondria, and centrioles. Let's start with the endoplasmic reticulum. That's the little network within the cytoplasm. Rough endoplasmic reticulum, which I mentioned earlier, are studded with ribosomes that produce phospholipids and proteins of plasma membrane. Produces proteins for secretion and for lysosomes, so basically more protein. Endoplasmic reticulum, especially the smooth, they help detoxify the cell, and cells that synthesize steroids as well. So the endoplasmic reticulum is a winding network of thin membrane sacs found in close association with the cell nucleus. The smooth and rough endoplasmic reticulum are very different in appearance and function. Rough endoplasm is studded with numerous ribosomes, which are sites of protein synthesis, whereas smooth endoplasmic reticulum synthesizes phospholipids, lipids, steroid hormones, and regulates the concentration of cellular calcium. Also metabolizes some carbs and breaks down certain toxins. So the smooth endoplasmic reticulum is actually very, very important. Here's a good graph of the difference between a rough and a smooth. And basically, if you look at a rough endoplasmic reticulum, it's going to have all the little spots right here, which are what? Ribosomes. And then the smooth, if you look right here, does not have the ribosomes on there. So that's the difference between a rough and smooth. Ribosomes, locations, where are they found? They're found in the cytosol. They're found on rough endoplasmic reticulum. They can be found in the nuclear envelope. They're found in nuclei, and they're found in mitochondria. So the proteins are found all right here. Now, what does messenger RNA help do? It assembles amino acids, which are like the building blocks for proteins. And we'll talk a little bit more about amino acids and proteins and all that stuff when we do the digestive system. So this is just a brief introduction. I'm going to dive into more detail about a lot of this stuff, but you don't want to get too detail-y in the first. Remember, you got to look at the big picture first. Look at the big picture and then study the specific things. I think sometimes as students, we try to concentrate on the real nitty-gritty, the real specific stuff, when you realize, well, wait a second. What am I even studying here? Oh, the human body, and then go back into that. So that's my tip of the day, is try to look at the big picture first and then concentrate, right? If you look at your whole life, maybe you got a B on one of the classes, or if you got a B on one paper. Who cares, right? That's just one small aspect of your life. If you look at the overall life, and if your overall career or overall school, you're fantastic. You're fine. Don't worry about it. You are all smart. Everybody has their own ways of portraying that. So don't ever let anybody tell you any different. You can all be successful. Now let's look at the Golgi apparatus. Remember I told you that was the UPS of the cell? So Golgi apparatus manipulates products from the rough endoplasmic reticulum and also produces new organelles called lysosomes. Remember lysosomes? Those are the little Pac-Man. Proteins and other products of the endoplasmic reticulum are sent to the Golgi apparatus, which organizes, modifies, packages, and tags them. So you might get something from the ER and say, hey, do something with these. Well, they're like, okay, well, I'll package it and I'll ship it out of the cell. And that's the job of the Golgi apparatus. So enzymatic proteins are packaged as new lysosomes or packaged and sent for fusion with existing lysosomes. So here's a lysosome. Enzymes in a single unit membrane. They clean up the cell. That's the key right there. Lysosomes help clean up the cell. Autophagy, which is breaks down organelles, but apoptosis, which is programmed cell death. That's very interesting, right? Apoptosis, which is programmed cell death. So if you think about it, I'll give you an example of this a little bit later on. But phagocytosis, which is, okay, there's a food particle here, then it engulfs it, and then it brings it inside here. But then we're say, okay, we're done with this. How do I get out of this? And they'll have a little vacuole and spit it back out. So the lysosome is what's going to say, okay, let me take in stuff that doesn't need to be in the cell membrane or the cytoplasm, and I'm going to try to break it down a little bit, and I'll spit it out into smaller little pieces when it doesn't make sense. So it helps clean up the cell. Okay. Now, remember I always told you that, okay, what is normal? You want to learn what normal is so you can learn abnormal, and throughout the semester I'm going to try to give you clinical examples of why all this stuff is important. It's one thing to memorize it, but why is it clinically important? Well, let's say now you know what a lysosome does. Let's say something goes wrong with lysosomal storage. So lysosomal storage diseases result when one or more lysosomal digestive enzymes are mutated and do not function properly. And guess what it's called? It's called Tay-Sachs disease. And Tay-Sachs disease is a condition in which the patient lacks a lysosomal enzyme needed to break down glycolipids in the brain cells. So if you can't break down glycolipids in brain cells, guess what? The glycolipids build up as a result of this defect, interfering with the nervous system functioning. It's usually seen predominantly in infants of Central European Jewish descent. Unfortunately, it can cause seizures, mental retardation, blindness, and, this is sad, death before the age of 5. So a lot of these diseases, you're thinking, why, and understanding how they come about and the physiology behind it is important. Some of these diseases are autoimmune. There's no cure. There's a lot of diseases, unfortunately, that we don't really understand, and unfortunately, we don't have a cure for. And as science improves, hopefully we can get down that path. But as far as right now, there is no cure for Tay-Sachs disease. Peroxisomes, they usually resemble lysosomes. They oxidize organic molecules, especially fatty acids. They produce hydrogen peroxide, oxidize other molecules, excess broken down to water and oxygen by catalase, and they're abundant in the liver and kidneys. Remember we talked about cholesterol in the plasma membrane? Well, your body, the human body, produces all the cholesterol it'll ever need, and guess who produces it? The liver. So really, you don't need to eat animal products which have cholesterol in order for you to survive, because your liver will make all the cholesterol it needs. So those that eat too much meat, red meat and stuff, you have an excess of cholesterol, and that's what it is, but because your body produces all the cholesterol it will ever need. Now, some of you will produce too much cholesterol, and that's a genetic component to it. Here's a peroxisome. Again, remember it resembles a lysosome. Peroxisomes are membrane-bound organelles that contain an abundance of enzymes for detoxifying harmful substances and lipid, which is fat, metabolism. Oh, the good old mitochondria. We love the mitochondria. The mitochondria are energy conversion factories of the cell. A mitochondria is composed of 2 separate lipid bilayer membranes. Along the inner membranes are various molecules that work together to produce ATP, the cell's major energy currency. Centrioles, these are very important in mitosis. They have microtubules, 9 groups of 3. There's a centrosome, cytoplasm that contains perpendicular pair, cellular division, and basal body. Now let's see if I can show you. Well, when we do mitosis, I'll give you a little bit more detail about the centrioles. But let's talk about the brain of the cell. You guys ever remember the cartoon Pinky and the Brain? I used to love that cartoon. They're like, are you pondering what I'm pondering, the brain? And it's like, what is that? To take over the world. Oh, these cartoons. They don't make cartoons like they used to. Now they have weird cartoons. I used to love the old school cartoons. Remember the old school cartoons? What are they? Voltron, Thundercats. You're like, oh God, Patel, you're old. Yes, I am. But I'm trying to enlighten you and teach you some of the old ways as well. I'm going to give you some 80s and 90s music later on as well. Alright, so here's the nucleus. It's the control center of the cell. The nucleus of living cells contains the genetic material that determines the entire structure and function of the cell. So inside the nucleus, you're going to have chromosomes. The human body has 46 chromosomes, 23 from mama and 23 from papa. That's why you look a little bit like your mom and a little bit like your dad, a little combination. Now, sometimes you look a lot more like your mom and sometimes you look a lot more like your dad. All right? Well, it depends on how much dominance and how much genetic information was passed on during mitosis and meiosis. Okay? So we'll talk about that, the difference between mitosis and meiosis. Inclusions, well, inclusions are not essential to cell survival. They have stored cellular products, pigments, fat droplets, granules of glycogen, foreign bodies such as dust particles. Ooh, we don't like these foreign bodies of viruses and intracellular bacteria. We need to get rid of those. So inclusions, not essential to cell survival. What is the cell cycle? Well, here's mitosis in a nutshell. Basically, it goes through different cycles. The way that I like to remember mitosis is intelligent people make awesome tacos. Intelligent people make awesome tacos. So you're going to have to learn these acronyms throughout the semester and say, okay, how do I memorize all this stuff? Patel's having me memorize too much stuff. I'll start making acronyms, associate things, and make a story, and you'll remember stuff. Remember, you're going to have difficulty memorizing all the muscles, the origin insertions, but try to make a story out of it. That's how I remember a lot of things. How do I associate? I have hundreds and hundreds of students every single semester, and like, how do I try to remember their names? I associate, and I say, okay, well, this person likes to play basketball. This person likes chocolate. This person is from this and this. And so you associate, and you'll remember their names a little bit easier. So in order to memorize material, you're going to have to make stories or acronyms. So let's start with interphase. So interphase is usually the cell, that's most of the cell cycle. Okay? So mitosis is only a small part of the cell cycle. And what happens is these different, gap 1, which is the G1 phase, the cell grows. Right? The cell is going to grow. Then the S phase, the DNA synthesized by, so the cell duplicates its DNA. So remember, you have 46 chromosomes. Well, during this time, the centrosome is going to duplicate, and you're going to make 46 times 2. You're going to make 92 because we have to separate. We have to make identical copies. So we have to go from 46 to 92. G2 phase, or the gap 2, cells grow some more, so you're going to continue to grow. Okay. Then what's going to happen is the prophase, chromatin condenses into chromosomes. So here's the intelligent people. Intelligent people. That's phase 1, prophase. Chromatin condenses into chromosomes. Nucleolus disappears. Okay. Then you get a pro-metaphase. So that's the phase that goes right before metaphase. And during metaphase, what you should see is that the chromosomes line up along the metaphasal plate, which is an imaginary plane right here. So you have 92 chromosomes lined up here. So you're going, cells growing. You're going to start duplicating your chromosomes from 46 to 92, because I say I'm going to make an exact copy of you. So in order to make an exact copy, I need to go from 46 to 92. If I split the cell into two, well, I'm going to go 23, and 23 is not a human being. Okay, so you need at least 46 chromosomes to be a human being. All right, then the next phase, go from metaphase, you go to anaphase, where the chromosomes break at centromeres and the sister chromosomes move to the opposite ends of the cell. Right here. And then once they do that, what's going to happen is this cleavage furrow is going to start to form. So it's going to invaginate on itself. It's going to try to go in. And then during telophase, the nuclear membrane reforms. So you get a nuclear membrane. The chromosomes unwind into the chromatin and they start to go. So you have 46 here and 46 here. Okay. And then what will happen is cytokinesis is the actual pinching off, and that is division of the cytoplasm. So the definition of mitosis is division of the nucleus, but division of the cytoplasm is actually called cytokinesis. So remember that. And then there you go. Then the cell, the resting phase, cell leaves the cycle, and then it's going to go back and start that cycle. So that's mitosis in a nutshell. So let's go over it in more detail. Cell cycle interphase, that's the first gap phase, G1, that's growth and normal metabolic roles. So most of the cell cycle is right here. It's growing, it's going about its normal metabolic roles. Then in the S phase, the DNA start to replicate. We're going to go from 46 to 92. That's the synthesis phase. The the G2 phase, which is the second gap phase, that's growth and preparation for mitosis, and it's going to proofread the DNA. Say, hey, you know those exact copies that I made, the 46? Well, I'm going to make an exact daughter cell, so it better be the same. So in G2 phase, you get DNA proofreading. And imagine this, you make billions and billions of cells, and then billions and billions of cells go through mitosis, and they constantly are dividing, and it makes almost zero errors. There might be a little error in here, but some of them are negligible. But think about that. What a fantastic machine that you have. All right. During the M phase, which is the mitotic phase, that's the division of the nuclear material. So really mitosis, the definition of mitosis is division of the nucleus. So in prophase, the chromatin condenses, the nuclear envelope breaks down, nucleolus disappears, the spindle fibers form to connect into the kinetochore. Metaphase, the chromosomes align at the center of the cell. The aster attached to the plasma membrane, so you see that right on the imaginary line here. You got the chromosomes, the 92 of them lined up. The anaphase, the daughter chromosomes move to the opposite poles. And the telophase, the chromosomes at each pole to recondense, surrounded by the nuclear envelope, and the nuclei form again. Intelligent people make awesome tacos. Make sure you know what happens during each phase and you're able to recognize it for lab. So lab versus lecture. A lot of times people say, what's the difference? Well, lecture, you actually need to know the philosophy, the physiology, why it's occurring. Where the lab, you just need to point it out. So lab, for example, in lab, you would say, okay, this cell is going through metaphase. Can you recognize that on a microscope? Can you recognize that on a model? In lecture, you need to tell me what's happening in metaphase. So that's how you would study the difference between a lecture quiz and a lab quiz. Cytokinesis, remember the definition is division of the cytoplasm, begins in anaphase, the cleavage furrow develops, the cell pinches into two identical daughter cells. Here's something that's interesting that the Dalai Lama said. The Dalai Lama, when asked what surprised him most about humanity, he answered man. Because he sacrifices his health in order to make money. Then he sacrifices money to recuperate his health. And then he's so anxious about the future that he does not enjoy the present. The result being that he does not live in the present or the future. He lives as if he is never going to die, and then dies having never really lived. And this goes back to, the first day of class, and a lot of you are stressed, a lot of you are anxious, and you're pressuring yourself to get good grades in this class. And yes, good grades are important. And I am really, really, really happy that you're choosing a career that puts a lot of pressure on you, but that's not what you want at the end of the day. Okay? Can you learn the material? Can you be the best healthcare professional you can be and help that person? At the end of the day, so regardless of what grade you get in this class, I know all of you want an A, and if you work hard, you'll get an A. But even if you get a B, if you learn a lot of the material, why the human beings work, how the human body works, how can you explain it to a patient, no one's ever going to ask you what grade you got in here. Okay, so when you go to an interview for let's say PT, OT, and say, oh, yes, such and so, oh, I see you got a B in anatomy. And I was like, well, I understand that my grade doesn't reflect, but I learned a lot in anatomy, and I can help that person. I'm empathetic. And so again, be charismatic, be empathetic, and you'll carry you a long way. No one will ever ask you what grade you got in this class. Okay? You just have to act the part. But I want you to learn the material as if you're going to help your grandma or your family member. So put some time and effort into it. And if you put the time and effort into it, guess what? You'll get an A. I don't give grades. You earn the grades. That's the common theme. So I don't want any emails saying, I need an A. You don't need an A. All you need is to learn the material. All right, here's a good example of the cell cycle. The two major phases of the cell cycle includes mitosis and interphase, when the cell grows and performs all of its normal functions. Interphase is further divided into G1S and G2 phases. Okay. So if you look at this, this is all interphase. And really, mitosis is only a small part of the cell cycle. Crazy, right? This is all interphase. Intelligent. People make awesome tacos. That's mitosis right there. So a lot of times people think that, oh man, cells are always going through mitosis. No, they're actually going through interphase most of their life cycle. So here's mitosis. What happens at interphase? DNA is replicated, cell prepares for mitosis. Prophase, chromatin condenses, making the chromosomes visible. Here's prometaphase, nuclear envelope breaks down. Here's metaphase, the chromosome become aligned. Anaphase, the sister chromatids separate and the resultant daughter chromosomes move toward the poles. And telophase, daughter chromosomes reach the poles and form two nuclei. Okay, so this is just a good summary of what we talked about as far as mitosis is concerned. Here's another chart that you can look at. Okay, and you're thinking, wow, there's a lot of repetition. That's right, anatomy is all about repetition. The more you hear it, the better it's going to be. Remember, it takes the human brain probably a minimum of 7 times to meet someone before you remember their name. So looking at this at least 7 times is important. And we'll talk about how your body and your brain remember information. Because after this lecture, guess what? 50% of it in a few minutes will be gone. That's right. You're not going to remember half the things that I told you. That's true, because the brain just doesn't work like that. The only way that it keeps remembering is repetition, repetition, repetition. Okay, and that doesn't mean repetition for hours on end. Repetition here a little bit, then taking a break, go and review it again a little bit later, then the next day. But if you're only going to review this right before a quiz, you won't do well. So try to spend a little bit of time every day and look over the material. And that's how you'll do well. That's how you get an A in this class. All right, let's talk about stem cells to end it. Stem cells are immature cells that can develop into one or more types of cells, specialized cells. Developmental plasticity. So stem cells are great because they can develop into any cell. Adult stem cells in most body organs produce cells for normal turnover. They're multipotent, for example, bone marrow cells. But then you have these embryonic stem cells, which the research loves. They're embryo up to 150 cells. Pluripotent. They can go into multiple and excess of in vitro fertilization. So the reason that we like embryonic stem cells is we can convert these embryonic cells into liver cells, heart cells, etc., etc. Adult stem cells, they'll just found in the most body organs, and they'll just make the the same cells that they belong to the organ. But we like these embryonics for research, cancer research, to make other kinds of cells. So how does it work? A stem cell, essentially the building block of the human body. Stem cells are capable of dividing for long periods of time, are unspecialized, but can develop into specialized cells. The earliest stem cells in the human body are thus found in the human embryo. The stem cells inside an embryo will eventually give rise to every cell, tissue, and organ in the fetus's body. Unlike a regular cell, which can only replicate to create more of its same kind, a stem cell is pluripotent. When it divides, it can make anywhere one of the 220 different cells in the human body. Unreal, right? So that's why stem cells also have the capability to self-renew. They can reproduce themselves many times over. So that's why stem cells are important. All right? The end.